we were really interested to find out whether we could extract animal DNA from air samples as a way of non-invasively monitoring biodiversity. And the actual experiment we conducted was to look at whether or not within a zoo environment where the animals are confined, we know where they are spatially, but also we know exactly what they are, whether we could recover that measure of biodiversity. So we sample species in an area and then match what we found to what we know was supposed to be on site. And the net outcome was actually very successful. We recovered quite a lot of the species in the zoo, those that we targeted, but also species that we knew were there but hadn't directly been sampled. And we also got some evidence of the wildlife in the area, amongst the more interesting European hedgehogs, which are critically endangered in the UK. We also sampled the diet of the animals. So sometimes we were able to detect what the animals had recently eaten from the air, probably from things like aerosolized feces and remembrance of their dinner that were floating around in the air. We were able to collect them as well. Well, we recovered evidence for mostly mammals, which was our target. There were some birds that we also sampled as well. Lots and lots of lemurs, binturongs, dingoes, tyra. There was some tiger DNA that we were able to collect in the area. It's based on what was actually at the zoo, but we also got things like zebra finches and budgerigars and some of the cranes that they had on site. We were able to sample their DNA as well. We were really looking to see whether we could use this for mammal surveys. I'm a mammologist by training, so I was interested in new ways we could track terrestrial mammal biodiversity. But what we found suggests that this has much wider applications. At this point, we can't quantify. I couldn't tell you how many hedgehogs are in the vicinity. It's more of a presence-absence test. Are they here on site? But that alone is very, very important when you're trying to sample for things that are critically endangered. You want to know if they're present in an area, whether that area itself should be protected.